Imagine a life irrevocably shaped, its trajectory altered by a single, seemingly insignificant misplaced letter. A tiny error, a minuscule deviation from the norm, holding the power to dictate destiny. This letter resides deep within the very blueprint of your being, in your DNA. It's a silent saboteur, working tirelessly against the natural order. For millions grappling with the harsh realities of genetic diseases, this isn't a hypothetical scenario. This is their everyday reality. A constant battle against an invisible foe, a relentless struggle for normalcy. Take, for instance, sickle cell anemia, a debilitating condition that disproportionately affects certain populations. This devastating illness is caused by one tiny, almost imperceptible error in the gene responsible for producing hemoglobin, the protein in red blood cells that carries oxygen. This single, solitary change, this minute alteration, warps the normally round and flexible red blood cells, transforming them into rigid sickle shapes resembling crescent moons under a microscope. These misshapen cells cause excruciating pain, progressive organ damage, and a lifelong cloud of uncertainty hanging over those affected. It's a relentless, daily struggle against the very fabric of your own biology, a constant battle against your own body. For generations, medical science has been limited to merely managing the symptoms, providing temporary pain relief, administering life-sustaining blood transfusions, and offering various supportive therapies. But until recently, we've never possessed the tools to truly fix the root cause of the problem, the underlying genetic typo itself, the fundamental error in the code of life. The audacious idea of correcting that single errant letter, of rewriting the genetic code, once seemed relegated to the realm of science fiction, a distant dream beyond our grasp. But now, with the advent of revolutionary technologies like gene editing, a new era has dawned, offering a tangible way to rewrite the very code of life, to correct the errors that lead to so much suffering. Instead of being limited to just treating the symptoms, masking the pain, and managing the complications, we can now dare to imagine curing the disease at its very source, eradicating it from the genetic blueprint itself. This is the powerful promise that drives dedicated scientists worldwide. Not just incremental improvements in treatment, but the potential for true, lasting cures. Sickle cell anemia is just one poignant example. Thousands upon thousands of other diseases are caused by similar single-gene mutations, each representing a unique challenge and a potential target for gene editing therapies. Each disease represents countless families and tight-knit communities, all united in their fervent hope for a life-altering breakthrough, a chance at a healthier future. The quest for precision gene editing is more than just a scientific endeavor. It's a deeply human endeavor, fueled by compassion and empathy, driven by the profound desire to alleviate suffering and rewrite the narrative of diseases that are currently written into our very DNA. This is the profound and deeply meaningful why behind the science, the driving force that propels us forward. Imagine having the power to rewrite the very blueprint of life. What tool lets us edit the code of life with such precision and control? It's called CRISPR Cas9, a revolutionary technology that's changing the landscape of genetic engineering. Think of it as molecular scissors, but not just any scissors. These are guided by a programmable GPS, allowing them to target specific locations within the vast expanse of our DNA. CRISPR has two main parts that work in harmony: a guide RNA and the Cas9 enzyme, the molecular workhorse that does the cutting. The guide RNA is like a search query entered into a powerful database, meticulously designed to find a specific DNA sequence within the genome. It acts as the address that the Cas9 enzyme needs to locate. Cas9 is the scissors, the cutting tool, ready to cut the DNA at the exact spot the guide RNA leads it to. It waits patiently, ready to be deployed with pinpoint accuracy. Together, they scan the genome, tirelessly searching for the sequence that matches the guide RNA's instructions, find the target, and make a precise, clean cut in the DNA. What's truly fascinating is that this system wasn't invented in a lab by human ingenuity. It was discovered in bacteria, nature's own tiny chemists. Bacteria use CRISPR as an immune system, a defense mechanism against viral attacks, storing snippets of viral DNA like keeping mugshots of past enemies. This allows them to recognize and destroy future invaders with the same genetic fingerprint. Scientists, with their insatiable curiosity, realized we could harness this system, this natural marvel, for our own purposes. By designing custom guide RNAs tailored to specific sequences, we can target any gene we choose with incredible accuracy. Delivering the guide RNA and Cas9 enzyme into a cell, like sending a targeted delivery package. lets us cut the genome wherever we want, opening up possibilities previously confined to science fiction. This precise cut is the first step, the crucial incision, in editing life's code, allowing us to rewrite genetic destiny. CRISPR's elegance lies in its simplicity and adaptability, making it a powerful tool in the hands of researchers and clinicians. It's a natural defense mechanism, honed by evolution, now repurposed for medicine, offering new hope for treating previously incurable diseases. With CRISPR, we can target faulty genes at their source, correcting the errors that lead to devastating illnesses. The possibilities for treating genetic diseases are unprecedented, offering a glimpse into a future where genetic ailments are a thing of the past. This is the foundational magic, the core principle of CRISPR Cas9, a technology that holds the key to unlocking a new era of genetic medicine. The world of genetic engineering is rapidly evolving, and at its heart lies the incredible precision of CRISPR technology. Making a precise cut in DNA, a seemingly small act, is the key that unlocks a world of possibilities. This precision opens the door to real cures, offering a beacon of hope for diseases that were once considered incurable. It's a new chapter in medicine, where we're not just treating symptoms, but targeting the very source of the illness. After Cas9, the molecular scissors, precisely snips the faulty gene. The process of correction begins. It's a targeted strike against the genetic defect, paving the way for the cell's natural mechanisms to take over. Once the cut is made, the cell's repair systems take over, initiating a cascade of biological processes. These systems are designed to mend the broken strands, restoring the integrity of the DNA. Sometimes, instead of repairing, the cell disables the gene, a useful strategy for stopping harmful proteins from being produced. This is particularly valuable in cases where the gene is actively causing damage. But the real breakthrough, the true game changer, is in precise repair. This is where CRISPR's potential truly shines, offering the possibility of correcting genetic errors with unparalleled accuracy. If we provide a correct DNA template, a blueprint for the desired sequence, the cell can use it to fix the error. The cell essentially copies the correct sequence from the template, replacing the faulty one. It's like pasting over a typo with the right word, correcting a mistake in a document. This simple analogy captures the essence of gene editing, identifying and correcting errors in our genetic code. This means we can rewrite mutated genes, effectively reversing the effects of genetic disorders. It's a revolutionary concept that holds the key to treating a wide range of diseases. Imagine replacing the sickle cell mutation, the genetic flaw that causes so much suffering. This could eliminate the painful crises and debilitating complications associated with the disease. Or fixing cystic fibrosis, a devastating lung disease caused by a faulty gene. Gene editing offers the potential to restore normal lung function and improve the quality of life for those affected. And more, the possibilities are vast and expanding every day. The ultimate goal is a permanent, one-time cure, a lasting solution that eliminates the need for ongoing treatment. This would be a monumental achievement, freeing patients from the burden of chronic illness. This is achieved by correcting the genetic blueprint, addressing the root cause of the disease at its source. By targeting the underlying genetic defect, we can potentially prevent the disease from ever manifesting again. CRISPR's promise extends to thousands of inherited conditions, offering hope to families affected by these disorders. From rare genetic diseases to more common inherited conditions, the potential applications are immense. Imagine halting Huntington's disease, a devastating neurodegenerative disorder. Gene editing could potentially prevent the progression of the disease, preserving cognitive function and quality of life. Or restoring muscle function in Duchenne muscular dystrophy, a debilitating muscle-wasting disease. Gene editing could potentially repair the faulty gene, allowing muscles to regenerate and improving mobility. We're on the threshold of a new era, a revolution in medicine driven by the power of gene editing. This is a time of unprecedented opportunity, where we can finally target the root causes of disease. We are addressing the root causes of disease, not just masking the symptoms with temporary treatments. This approach offers the potential for long-term solutions and lasting relief. For families, CRISPR offers the hope of ending inherited suffering, breaking the cycle of genetic disease. It's a chance to rewrite their family history, ensuring a healthier future for generations to come. This is the life-changing promise of gene editing, a future where genetic diseases are no longer a life sentence. CRISPR is set to make medicine truly personal. Traditional medicine treats the average patient, but our genes make us unique. With CRISPR, therapies can be tailored to each individual's biology. In cancer, for example, we can engineer a patient's immune cells to target their specific tumor. Living medicine made just for them. For infectious diseases, CRISPR could recreate protective mutations, like making immune cells resistant to HIV. CRISPR also enables rapid, precise diagnostics, detecting infections or cancer in minutes. Imagine instant, personalized treatment decisions based on your genetic code. Medicine will no longer be generic, but designed for you. This is the dawn of truly individualized healthcare. The CRISPR revolution is already underway. In clinical trials, patients with sickle cell disease have had their blood stem cells edited to produce healthy hemoglobin. People like Victoria Gray have gone from constant pain to living symptom-free after a single treatment. This is proof that CRISPR therapies can be safe and transformative. Another breakthrough, treating genetic blindness by injecting CRISPR directly into the eye, restoring vision for some patients. These early successes pave the way for more conditions to be treated. Each trial teaches us how to deliver CRISPR safely and effectively. The courage of these first patients is lighting the path for millions more. We're witnessing the first steps of a medical revolution. The future of gene editing is happening now. With great power comes great responsibility. CRISPR's promise is matched by its risks, like off-target effects, where the wrong gene is cut. Scientists are developing safer enzymes and smarter guide RNAs to minimize mistakes. Delivery is another challenge. Getting CRISPR into the right cells, especially in solid organs, requires new solutions. Building safety into every therapy is essential. The scientific community must be transparent and ethical, working with regulators and the public. This isn't a race, but a careful journey. The power to rewrite life's code demands humility and stewardship. Beyond science, CRISPR raises profound ethical questions. Somatic editing treats one person and isn't inherited, widely accepted as medicine. Germline editing, which changes eggs, sperm, or embryos, would alter future generations. The potential to eliminate inherited disease is powerful, but so are the risks. Where do we draw the line between therapy and enhancement? Could gene editing deepen social divides or have unforeseen consequences? These are questions for all of society, not just scientists. We need global dialogue, clear regulations, and shared values. The decision to edit the human germline is one of humanity's greatest responsibilities. The next decade will transform medicine, led by CRISPR. Cures for diseases like sickle cell will become routine, not rare. We'll move from managing illness to curing it, tackling even complex diseases like heart disease and Alzheimer's. CRISPR, combined with AI, will accelerate discovery and personalize care. A genetic diagnosis will no longer be a life sentence, but the start of a cure. The hospital of the future will deliver precise, curative genetic medicines. This future requires innovation, ethics, and global access. The power to edit our biology brings hope for less suffering and a deeper understanding of what makes us human.